and welcome to the channel. Met Cancer, Metabolic, not genetic. My name's Vincent and you can find me on X at Vincent Gledhill, which amazingly is my name. Now, sorry I haven't posted any content for a while. I've had to get a job to pay the bills. I've made this video so that you can share it with anyone that doesn't believe that cancer is a metabolic disease. It is not, and I repeat, not a genetic disease. Here is the definitive proof from Professor Thomas Seyfried, and all of the papers discussed in this video I have linked in the description. So, I give you Professor Thomas Seyfried.
Today I'm going to talk a little bit about how cancer develops and what could be responsible for cancer. Let's talk first, what is cancer? It's dysregulated cell growth, cell division out of control. How does this happen? What's underlying cells in a particular tissue or organ of our body to start growing out of control? Well, if we go to the National Cancer Institute web page, it says, how does cancer develop? And what it actually says is cancer is a genetic disease. That is, it's caused by changes to genes that control the way our cells function, especially how they divide. So cancer is caused by certain changes to the genes. What are these changes? How they're expressed, how they function, and why they go awry is considered mutations in these genes that could come from any number of provocative agents from the environment or diet and lifestyle or things like this. Other pages from the National Cancer Institute say cancer is many, many different diseases. Colon cancer, breast cancer, bladder cancer, brain cancer. We have different kinds of mutations that might be present in some of these different cancers, although there are some commonalities to the mutations that you find. But obviously there's going to be differences in genetic expression profile, mainly because all of these tissues have their own unique genetic architecture. But when we look at cancers, do all cancers have mutations? Do all mutations cause dysregulated cell growth? These are some of the things that we have to look at. So there are a significant number of papers showing that some cancers have not been found to have mutations. So we have the work of Parsons in 2008. If you look at their table 3, you will find that one of the samples, the BR20P sample in table 3, was not found to have any mutations in the 3 major signaling pathways, the P53 pathway, the PI phosphatidylinositol 3 kinase pathway, and also in the retinoblastoma pathway. And it is interesting when you find a piece of evidence that should not be according to the somatic mutation theory, which says that cancer is a genetic disease based on somatic mutations. You find a sample that has no mutations. How do you explain that? And what's often done is you ignore it, and that's exactly what the authors of that paper did. They did not discuss it. The paper by Greenman and Michael Stratton's group from England clearly described a number of breast cancers that had no mutations. Majority of breast cancers had thousands of mutations, but using their technology, they were not able to find mutations in some of the breast cancers. And again, these should be red flags, inconsistent with the somatic mutation theory. And yet they're ignored. So if you see something that's inconsistent with your theory, the first thing that a scientist would do would try to explain this in light of the theory that drives the research. Work from Versteeg also described certain cancers with no mutations, especially in the ependymoblastoma, childhood brain tumor, are known. This is a very aggressive childhood brain tumor, but yet some types, some subcategories of these tumors have no mutations, yet cells are dividing out of control. Dysregulated cell growth in the absence of gene mutations are data inconsistent with the somatic mutation theory. Yet these inconsistencies are essentially ignored or not discussed for reasons that aren't clear. I think competent scientists should be compelled to question observations that are inconsistent with the central theory. The persistence of thinking cancer is a genetic disease when we have clear evidence that in some cases it cannot be, and yet the National Cancer Institute supports all kinds of research for cancer being a genetic disease. All of this leads in part to the persistent death rates of cancer. 1,700 people a day in the United States dying from cancer, which is about 70 an hour. This is all supported by the evidence coming out from the American Cancer Society. If the theory is wrong, and we keep studying and developing therapies based on an incorrect theory, we will not get any major advance. Now, and then of course, we have driver mutations. So it turns out that not all mutations are considered pathological in the sense of contributing to dysregulated cell growth. Rather, we've defined some genes as being especially important for driving dysregulated growth, and that's called driver genes. And at first, people thought, well, okay, not all mutations are involved in dysregulated cell growth, only certain called drivers. Drivers drive dysregulated cell growth. Well, now we find new information from a number of researchers, Martin Carina, Yokoyama, Yitzhak, Martin Jimenez, all recent, showing massive numbers of driver mutations in normal tissues of people's bodies that have no dysregulated cell growth. How do we explain cancers with no mutations and normal cells and tissues with loads of driver mutations that never become cancer? These observations are absolutely inconsistent with the somatic mutation theory and the statement on the National Cancer Institute website. How is it possible that they maintain these websites when information in the scientific literature does not support the information on the website? This is just, people need to question the people. Who's responsible for setting up the websites? Do they not read the scientific literature? Do they not understand the scientific literature? Why they choose to ignore the scientific literature? The most devastating evidence against the somatic mutation theory was the nuclear mitochondrial transfer experiments. These were done by a large number of scientists across various types of disciplines using different cancer models, coming to a similar conclusion that it's something in the cytoplasm, particularly the mitochondria, that are controlling the dysregulated cell growth rather than the mutations in the nucleus. The papers by Barbara Israel and Warren Schaefer from 1987 and 1988 clearly showed that when the nucleus of an aggressive cancer cell, when the nucleus of the tumor cell was placed in the cytoplasm of a non-malignant cell of the same genetic background, no cancer was seen, no dysregulated cell growth, despite the continued presence of the somatic mutations in that nucleus. On the other hand, when the nucleus of the normal cell, a genetically normal nucleus, was placed in the cytoplasm of the tumor cell, a significant number of tumors were developed from that construct. These are just the opposite of findings that you would have expected. You would expect a normal nuclear genome to repress the tumor growth, and we find just the opposite of that. I've published major papers on this. I've reviewed the evidence from these independent experiments over and over again. And what we constantly see is these evidences do not support the somatic mutation theory. And this was done also in vivo in different animal organisms. For example, the frog experiments done by McKennell and his colleagues in 1969, where there was a very aggressive kidney tumor in this frog called the luck frog. And the nucleus of the aggressive kidney cell was taken out and implanted into a freshly fertilized frog egg that had a normal cytoplasm. And what was developed was a completely normal tadpole in every way without any dysregulated cell growth. This should not have been found, because the tadpole, you could even cut the tail off the tadpole and it regrew a new tail, all directed by the nucleus of the tumor cell. These findings are inconsistent with somatic mutations leading to dysregulated cell growth. So when you put all this together, it becomes hard to imagine that any rational mind could continue to think that cancer is a genetic disease when you have this massive evidence to say it can't be. The consequences of the persistent belief that cancer is a genetic disease is over 600,000 people dying in the United States each year, which comes to almost 1,700 people a day, 70 an hour, continue to die from this disease because of an incorrect theory, which is driving the industry to produce therapies based on the somatic mutation theory. If the theory is incorrect, then we will not drop the death rate anytime soon. And I will present evidence that this is a mitochondrial metabolic disorder driven by a fermentation metabolism using two fuels, the fuel glucose and glutamine. In fact, our big paper just was accepted, clearly showing that glutamine is a fermentable fuel, as is glucose. So once people realize that cancer is a mitochondrial metabolic disorder driven by fermentation metabolism, making it their inability to burn fatty acids and ketone bodies, now we have a clear solution to this cancer problem that can allow people to live far longer with a higher quality of life. It just takes time for individuals to come, read the literature, understand the literature, and then come to their own decision, then question, why are we continue to persist with failed therapies based on an incorrect theory?